one's career happen, and sometimes the field greatly benefits from such events. We were fortunate, for example, that Skinner did not become a writer of fiction. I submit that this was also the case with Christopher Newland. Christopher Newland graduated in 1972, not about writing, but he graduated in 1972 from Auburn University with a degree in electrical engineering. He was a consulting engineer, and I assume he was making probably pretty good money when he decided that he wanted to go to graduate school to study experimental psychology. He attended the Georgia Institute of Technology, studied under Jack Ma. Upon receiving his PhD in 1982, he went to study with Bernie Weiss and Vic Ladies as a postdoc at the University of Rochester. In 1988, he joined the faculty in the Department of Psychology of Auburn University, where he has been ever since. His accomplishments are numerous and of exceptional quality. I will only highlight a few so that we can hear him speak. Chris's main interests are in behavioral toxicology and behavioral pharmacology, and in particular, mathematically based descriptions of behavioral processes. Chris has been successful in applying and translating experimental analysis of behavior into other arenas, such as environmental health sciences. Chris has had almost continuous funding since 1990 by such agencies as the National Institute of Drug Abuse and the National Institute of Environmental Health Sciences. He has over 50 peer-reviewed publications and book chapters. He is the current Vice President and Rising President of the Neurotoxicology Specialty Section of the Society of Toxicology. He is an editorial board member of several prestigious journals, including Neurotoxicology, Neurotoxicology and Teratology, and at JL. I could go on, but I must stop so we can hear Chris talk, which is entitled Applied Modeling and the Identification of Behavioral Mechanisms of Action, co-authored with Wendy Donlin, a former student and now at Johns Hopkins School of Medicine doing a postdoc. Please give a warm welcome to Chris.
Well, thank you very much, Mark, for that kind introduction, and frankly, I think we should probably stop now. Because, well, first I'm going to do something quite different. Before I do, I really want to introduce again Wendy Donlin, a co-author on this paper, because much of the work that I'll be presenting, especially at the end of the talk on Michelle's partitioning of behavior into bouts of responding, is actually due to her and was part of quite a lovely dissertation project. But today I'm going to do something a little bit different for a squad talk. I'm not going to be presenting a model, I'm not going to be defending a model or tinkering with parameters. Instead, I'm going to be talking about two particular models and how we've applied them, or exported them, or translated them into a different arena. I think for the rest of my life, this talk is going to be the defining moment of the phrase, it seemed like a good idea at the time. The reason is, there's so much to say about this, and there's so little. There's so much to say because the application of a particular model depends, there's a lot to say about this business because the application of a particular model to a particular application depends so heavily on a particular model and a particular application. There are many of those. And there's really relatively little to say for exactly the same reason. There aren't many general principles here. So be forewarned. Mathematical modeling is fun, but it's also quite useful. And its utility becomes especially apparent in the model when the model isolates critical determinants and relevant markers of behavior. In fact, that's nearly a direct quote from a nice paper by Mazur just recently. Models of operant behavior abound and have achieved impressive levels of sophistication along a number of dimensions. To distill behavior to its essential elements and to its influential variables, modelers have applied mathematics that are sometimes quite complex, and at other times remarkably simple, but not simplistic. Perhaps the latter category is simple, collection of models that I wish to emphasize here today, because they are especially suitable for export to other research domains. What, for example, could be simpler and more pregnant with possibility than the Manchin relation? In both a proportional and generalized Manchin form, this model captures an intuitive and straightforward understanding of the relationship between behavior and its consequences. Its essence could be explained to a third grader. We spend most of our time with the reward of greatest. Yet there are lovely subtleties embedded in the simple equation of response and reinforcement ratios that capture such properties as sensitivity to reinforcement, important biases, contextual control, response induction, and perhaps even motor characteristics of behavior. Even greater and more profound subtleties arise once it is recognized that even in steady state, choice is a dynamic and continuously adjusting process. Such an average is high a tug of war between the rich and the lean alternatives in their competition for an animal's behavior. More important, though, is what is staring us straight in the face. When we look at a Manchin function, that is the extraordinary amount of variability that is captured by ratio matching and its impressive consistency that is seen across a broad range of species. This shows Manchin functions that I've had some involvement with, generated from humans, working on computers, squirrel monkeys, working on levers, or my favorite, long-headed rat, working on levers, and even bluegill sunfish, a recent thesis by Kelly Bannon. How could we resist applying such a model? And in fact, it meets Pennypacker's criteria for technology transfer. Quantification, repetition, and verification. Now, I have a little more to say about this, but in brief, quantification. It provides quantitative descriptors of behavior, not just in independent and dependent variables, but also in terms of the model. Repetition. We can repeat this. We know what to do. And that gets to verification. We know what should arise when we arrange conditions in some sort of way. And if it doesn't arise, we know that there's a mistake. And that's important, I think, in exporting models and translating them. Surely this ability to predict and control behavior has some application, and in fact it does. The matching relation has been applied to substance abuse, agriculture, education, business, toxicology, to name just a few. And here lies the reason for modeling in the first place. Usually the development of a mathematical model arises when the behavior can be controlled precisely and described with a minimum of terms. This means that the phenomenon is fairly well understood, very well understood, in the sense that the conditions under which it appears can be described economically, quantitatively, and thoroughly. This knowledge can and should be exported to other domains. A recipe for selecting a model cannot be articulated, at least not by us, but it may be possible to identify some general guidelines in selecting a model. Before selecting the model, it's important to understand how it will be applied. Are the model's terms relevant to the proposed mechanism of action, for example? This seems obvious, but it's not always obvious. So this touches on the issue of validity. Quantifiability. Relevant and accessible dependent measures and independent measures of behavior should make contact with the terms of the model. I think the best models permit us to do this. And the final issue that's been important to me, for reasons that will become evident shortly, is scalability. Can the model be adopted not only to a different application, but to a large-scale application? And this makes contact with some important developments that are occurring in neurobiology, and that is the issue of behavior genetics. And considerations that fall under the rubric high throughput. I think we have an awful lot to bring to this table, and so far we haven't brought it, or at least not as much as we could. Okay, so to become concrete and to talk about our own approach. The problem that we have addressed here, and that we've been addressing in my laboratory for the number of years, is the behavioral toxicity of methylmercury. This is a neurotoxicant that could be of some interest to behavior analysts, in part because of the functional domains that are affected. When exposure occurs in adulthood, there's significant impairment of both sensory and motor function that can be traced to both central nervous system and peripheral damage, and that has significant behavioral consequences. To characterize effects, we've turned to a number of behavioral measures, including ones that I want to describe later, and that's high rate operant behavior. When exposure occurs while the brain is developing, methylmercury causes significant disruption in the development of the cerebral cortex. So let me unpack that a little bit. So it looks like we're losing a little bit of the slide, but I think that's okay. And I had a laser pointer. Okay. The modern era of methylmercury, our understanding of methylmercury poisoning actually traces back to an episode that occurred almost about 50 years ago, actually, in Smith and Smith's photo documentary. It traces back to the spring of 1956. But actually, the first appearance started with symptoms of the patient occurred earlier. But it traces back to a fission village in Minamata, Japan. This map actually is from Chiso Chemical, the company that actually dumped mercury in Minamata Bay, methylmercury in Minamata Bay. And it became apparent that there were two sets of symptoms. Chronic adult onset exposure, there were severe sensory effects, including blindness, severe motor effects. With congenital exposure, developmental exposure, there was mental retardation. There's relatively little mental retardation associated with chronic exposure, actually. But there was mental retardation and some more subtle sensory deficits. This was extraordinarily high levels of exposure, levels that are very difficult to experience today. But there are a couple of epidemiology studies that are being conducted, several, including two that are listed here, one in the Faroe Islands in the North Atlantic Sea, one in a resort community in the Seychelles Island off the east coast of Africa. If I had to choose, I think I picked the Seychelles, that describe very different exposure patterns, very different nutritional patterns, and different patterns of effects. In fact, effects that appear in the North Sea and not in the Seychelles. And this is one of the things prompting the work that we're doing in our laboratory, which is identifying developmental exposure to methylmercury paired with nutrition and sorts of nutrients that are found in fish. More on that later. So back to validity. So with methylmercury exposure, we have distinctions between adult onset and developmental exposure. When exposure occurs, when exposure occurs, it goes under significant impairment of sensory motor function that can be traced, as I said, to central nervous system damage. And when exposure occurs during development, the behavioral consequences are best identified and characterized with operant behavior. We've identified disruptions in the sensitivity of behavior of exposed animals to changes in reinforcement contingencies. And in fact, that seems to be one of the more sensitive endpoints. So we will talk a bit about choice and perseveration is also an issue in our laboratory, and I won't talk about that today. For reasons that will become apparent, we've turned to models of operant behavior. Okay. Selecting models for adult onset exposure is relatively straightforward. I think most people in this room can do that. For developmental exposure, the problem was a bit more difficult. And one of the reasons has to do, I think we understand now, has to do with the neurobiology of methylmercury effect. Excuse the slide from the textbook, but methylmercury seems to be a cortical neurotoxin. In adults exposed developmentally to methylmercury, you get a deranged cortical layering. And we know that the layering, this beautiful pattern of layering that occurs in the cerebral cortex is the result of a sequence of processes beginning with cell division, going through migration, differentiation, and so on. And these stages of development are especially sensitive to methylmercury exposure that occurs as it's developing. And this can give rise to both cerebral cortex and cerebellar cortices that are significantly affected. I also think development of methylmercury is sort of a neurochemical neurotoxicant because, and one where dopamine winds up being very important. And in part, this is probably related to the fact that the mesolimbic cortical dopamine pathway has its endpoint in the cerebral cortex. So in many ways, I think we're looking at the cortical neurotoxicant. Okay. From a behavioral neuroscience, so this is important in part because the old tried and true approaches to behavioral toxicology have not been very sensitive to methylmercury exposure. Tests of remembering, such as delayed master sample, delayed spatial alternation, various mazes, and so forth, have really not been very successful at identifying methylmercury, except at levels of exposure that are so high that they are of little practical interest. And in fact, in some cases, both in human populations and in developmental and in animal preparations, improvements have actually been seen in memory for reasons I can talk about later if you like, but I don't plan to here. From a behavioral neuroscience perspective, this failure of memory-based procedures to be sensitive to methylmercury is understandable, in part because these tests are so sensitive to hippocampal damage, and this region is spared by low-level exposure. Clues for a research domain to examine came from a study that we conducted that I was involved with a number of years ago with lead-exposed and 3 methylmercury-exposed squirrel monkeys. Commonly used fixed ratio and considerable schedules of reinforcement were somewhat helpful, but the main effect that was seen with the procedure that we decided to try, and actually it worked. The procedure came from thinking, I tend to approach these problems by thinking about the 3-term contingency, and I find this representation to be especially helpful. And really what we're focusing on here is more or less the response-reinforcer relationship. In the presence of choice, and to some extent with the partition and response frame, I think we're looking at this interaction. When we look at motor effects, I think we're focusing on the response. Those sorts of phenomena that fall under contextual control, including discrimination, sensory function, and memory, sensory function is a part of the picture. But discrimination and memory seem to be relevant to other neurotoxicants. So this slide represents a study that I was involved with animals in Lund University in Sweden, in southern Sweden. And what I have here are 3 data that came from 3 squirrel monkeys, a control monkey, a monkey exposed during gestation only to methylmercury, and a monkey that was exposed during gestation to lead. They were tested when they were several years old. The experiment went something like this. For a number of sessions, the animals worked on a concurrent schedule of reinforcement, in which half of the reinforcements were programmed to come from the left lever. After a number of sessions, 20% of the reinforcements were programmed to come from the left lever, and over the course of about 9 sessions, the control monkey behavior changed accordingly. Then we have other transitions later, to slightly over 80%, and 20%, and then 90%, and so forth. The mercury-exposed animals seem almost indifferent to this change in the reinforcement ratios. It just plowed forward. This is not an extraordinarily high level of exposure. The lead-exposed animal, this is an extraordinarily high level of exposure to lead, actually. The lead-exposed animal, his behavior went in entirely the opposite direction. We suspected we were onto something here. And we hypothesized that maybe what we're looking at is insensitivity to changes in reinforcement ratios. We tested that hypothesis with a behavior therapeutic intervention. Instead of 80% of the reinforcements coming from the right lever, 20% from the left, we had 99.9%, because we could do it instead, 99.9% of the reinforcements coming from the right lever. And with that, at least, we were able to move behavior around. So this suggested to us that at a behavioral level, what we're looking at is something to do with this response-reinforcer relationship. Okay, so these were the clues, and we have followed the choice workup, and we have expanded it to other areas. I want to say just a brief word about quantifiability and how it bears on the selection of a model. And actually one of those things is obvious once it's said out loud. The goal of the concurrent schedule studies was to look at sensitivity to reinforcement in steady state. One of the things that we discovered, though, was retarded transitions occurred when the reinforcement ratios changed. And what I haven't showed you yet is that in animals whose steady state levels looked just fine, it took them a longer time to get there. And so we began to modify the model to characterize transitions. And in 1990 or so, 1992, there wasn't a whole lot to go on. So we made it up as we went along. And with the shell model of response valve, The quantitative parameters that brought to the table for us was one parameter that described the motor component and one parameter that we think described the motivational component. But before we get back to that, I want to talk about this issue of scalability. And in order to do that, I have to tell you why this is an issue. So in order to study methylmercury in nutrition, we are exposing animals to methylmercury, some animals to methylmercury, 1 or 3 doses, and some animals to a diet that is rich in lean selenium or rich in lean omega-3 fatty acids. We are studying chronic adult onset exposure. We are also studying developmental exposure. So the issue arises here because group designs are necessary. The experimental analysis behavior has been notable for its ability to explain the behavior of an individual organism. I think that's why the models that come out of it are so useful. This is a strength both conceptually and methodologically. But as we translate our results, I don't think we need to rigidly adhere to all the methodological constraints that led to the development of the Operant principles in the first place. Instead, we need to be flexible, letting the question and the consideration dictate the means. For example, we're working with developmental neurotoxicant or a toxicant that when exposure is adult onset, the effects may be irreversible. The irreversibility may be handled with a small n design, although it's arguable whether such a design is ideal. With a developmental neurotoxicant, however, a pregnant animal is exposed and its offspring are followed. This makes all discussion of AB or ABA experimental designs moot. Our animals are not born again, even though we live in Alabama. Group designs are necessary. Control animals must come from different mothers and then exposed animals. And with different doses or nutritional interventions are required, and this increases the sample size even greater. So a typical experiment for us is a 2x3 experimental design where we have 0, a low, and a moderate, actually level of exposure to methylmercury, and a diet that is either deficient or sufficient in a particular nutrient. So 6 cells. Within each cell, we have different experiments. Because this is a developmental study, we have a lot invested in each animal, so we want to make the most out of it. So we follow the breeders for chronic adult onset exposure, and then rats have multiple animals in their litter. So one animal will be assigned to a drug challenge experiment, another animal to a high rate behavior experiment using a percentile schedule, and so on down the line. In order to achieve any experimental power, we're using 8 animals per cell, and I'm beginning to question whether that's enough. The ability to scale upwards from the troika of subjects that we like so much with experimental analysis of behavior to 48 animals then per experiment is crucial. So the type of experiment that that started me in this business and that keeps a whole lot of us entertained involves a handful of former teacher called Pythagorean design. And actually, I put pigeons there, but in fact, these matching functions came from bluegill sunfish, in which we used the classic EAB experimental design to get a handle on how to study this particular species, but that's just not going to work. 3 animals, each animal gives several performance measures. That's fine, but for our studies with 48 animals per group, we've got to have multiple sessions. Each session gives rise to multiple performance measures. Session 1, session 2, and the performance measure, session one sort of experiment, 48 animals, however many performance measures we have, we can't go in there with Excel and just tinker. So we connect our one computer running these animals with other computers doing the data crunching, and use the data management process. I can almost become a promoter of this. A data management software called RS1, which is one of those wonderful developments that makes it easy to study small n, to study individual subjects, and makes it hard to do group statistics. Has a lot of advantages. One of them is that I can write a program called Do a Day, which sucks in data from all of those subjects, tens of thousands of data points a day, generates 1,000 graphs, and this is thousands of graphs a day, which we are reading all, but we track them when we need to. And so this is the daily monitoring of current schedule performance, body weight. Of course, important to responding. For another procedure that I'm going to talk about later, multiple percentile DRH schedule, which we're looking at high rate behavior, the situation is a little more complicated. We have two different types of conditions. We have criterion responses and non-criterion responses. And one of the things that we started doing is keeping track of their response time distribution on a daily basis. So by looking at them this way, 10th percentile, 20th percentile, 30th percentile, and so on. And so the median response time for this particular condition is about 0.3 seconds, and so forth. And this allows us to track stability so that we can impose a different phase change, and we do all the things that we know how to do. Over on the right, these are derived measures that come from Shell's model. So we look at Bout length, Bout initiation rate, within Bout response rate, and so forth. And in order to produce one of these graphs, we have to do 4 multiple regressions for each subject, 48 subjects, and so forth. The fact that this model that I'll talk about more in detail later gives us such nice, straightforward, easy to interpret parameters has been enormously helpful for us. It is a robust, informative, and sensitive model, and very helpful in our hands. Okay, so we'll breeze through the matching models of choice and then spend the rest of the time talking about the other things. So I want to talk a bit about the rationale for incorporating this model and some of the extension of it. Oops, that's the wrong slide. Sorry. Okay. The rationale for developing the model of choice. We're interested in sensitivity to behavior to its consequences. I think we're studying frontal cortical function. Don't know for sure, but that's sort of a working hypothesis. The model is very well characterized. When we brought it into the laboratory, we knew exactly what to expect. It is a nice and robust model. It is highly conserved across species, so we were confident that what we were studying in one species would be informative about another species. The verification rationale was important because the monkeys and the people running the monkeys were in Lund, Sweden, and I was in Auburn, Alabama. Very different. And so and this was done in 1990 or so. There was a new thing called email and a new thing called TCP/IP that allowed me to actually log on over here. We take these things for granted now, but at the time it was quite a remarkable thing to have done, a fun thing to be involved with. But I was able to bring over data every day, monitor it, be sure things were going well, and that issue of verification, of knowing what to expect and whether what we got was what we expect was extraordinarily important. We couldn't do this otherwise. Okay. We have brought this procedure over to Auburn. Now we've been doing now single session transitions in long-lived transposed to methylmercury during development, and I'm not going to walk through much of that except to talk about another model that we've developed in our laboratory to describe choice and transition. Let me emphasize at the outset, this is an empirical model, nothing more. It is simply a description of the data, and it works very nicely, and it gives us parameters that are informative. So we build a logistic function to the acquisition of choice, where we go from 50-50, half the reinforcers coming from one side, to one in which, I forget what this transition is, probably a 4-to-1 transition. The logistic equation gives us a number asymptote, which describes where behavior ends up. It gives us a slope term, which describes this slope, which is roughly the number of reinforcements required to get from 1/3 to 2/3 of the transition, or 1/e to 2/e actually. And half maximal reinforcers, which is the number of reinforcers required to get halfway through the transition. I picked the logistic equation because it fits an S-shaped curve that we kept getting, because it gives us nice parameters, and also because it arises in population biology. And at the time I thought I had fantasy that it might lead to other modeling. Just very quickly, so this is what I just showed you, a control subject. These are methylmercury exposed subjects. In this particular case, the upper asymptote's not that different, but it took the animal 48 rather than 19 reinforcements to get there. And this is something that we see occasionally, not often, but occasionally in methylmercury exposed animals, the transition went in the wrong direction. We see exposure-related effects and transition size-related effects on the number of half maximal reinforcements required to get through a transition. So in control subjects, it takes about 20. At the time, I didn't believe it. I didn't think it would go this fast. Now that Baum and Davidson have published their choice and transition work, I'm wondering why it's going so slowly. But that's what does the data. And then the exposed animals, it took about twice as many reinforcers. Okay. Cruising for models. Part 2. We spent a fair amount of time examining choice and transition with these animals. But one of the other things that we wanted to do was develop some procedures for examining high rate behavior and another phenomenon that I'm probably not going to be able to talk about here today, response perseveration. So I want to talk a bit about the rationale for incorporating this partition analysis, the current application, the development and extension of the model, and we need to talk a bit about testing the assumptions of this model, because this model is not as well developed as the mentioned model of choice. And there are a couple of things that we wanted to probe in our own laboratory, and then briefly an application of the model. Once again, back to our old friend, the three-term contingency. Again, we are focusing on the response-reinforcer relationship. In one case, we know that with chronic adult onset exposure, we see some pretty significant motor deficits that occur. What we would like to do is determine whether high rate behavior could be an early indicator of exposure before we see a rather florid display of effects. With respect to perseveration, one of the things that we are interested in is what happens with persistence in the face of change. So, there's a poster that will be presented, I think it's Monday morning, with what we call the low rate challenge, in which we have high rate behavior that goes on for a long time, then we change things a little bit and require a low rate, and we look to see how quickly behavior changes. So, our first encounter with high rate behavior involved a procedure called differential reinforcement of high rate behavior. In part, this was driven by a very old study that indicated that this procedure might be particularly sensitive to developmental exposure to methylmercury. So we brought it into the laboratory and worked with it. We studied, the way that when you think about DRH schedules, it's not exactly the opposite of a DRL. DRL is nice to define. There are straightforward ways to define it. DRH is not quite the same. And so the way we brought it into the laboratory is we describe it this way. DRH21 simply means 2 responses have to occur within 1 second, 5 responses within 2 seconds, and 9 responses within 4 seconds. Each of these preserves an average inter-response time of about half a second. This worked nicely. The paper's been published with Aaron Rasmussen, and it identified some aging effects and some drug-related effects of methylmercury. But one of the things that we discovered as we were examining, as we were conducting this work, was that under a DRH schedule, there's a positive feedback loop that develops. Motor deficits decrease response rate. Okay? That's what we wanted. But this then decreases the reinforcement rate. If they're not making the criterion, then they're not getting the reinforcers. And if they're not getting the reinforcers, then you know what's going to happen next. There's going to be a further decrease in the response rate. But this isn't related so much to the motor deficit, it's related to that reinforcer effect. So we were interested in reinforcement contingencies that constantly adjust the response criterion according to recent performance. I started tinkering with titrated DRH schedules, and eventually we discovered a percentile schedule. One of those things that occurred to me while driving to work, taking a shower. And this is how we implemented this. So this is an attempt to generate high rate behavior while simultaneously disassociating performance from the overall reinforcement rate. So we defined a percentile schedule with 2 parameters, one that describes a look-back window, and one describes the proportion of IRTs that are reinforced. This is very new work. Some of this was just rate responding, short inner response time. This represents an event record with long inner response time. So I'm just distinguishing here short versus long inner response time. With our particular percentile arrangement, every response terminates a reinforcement. Terminates an IRT. We take that IRT, we compare it with the previous 10 IRTs and ask if it's longer or shorter than 50% of those IRTs. If it's shorter than those IRTs, then it's eligible for reinforcement. If it's not, it's not eligible for reinforcement. Here's one that's not eligible, here's one that is eligible. And so we're constantly adjusting we're reinforcing hydrated behavior while constantly adjusting the criteria according to what the animal studies. And this, look back on those slides forward, of course, for each response. These criteria in response time and a response times are then reinforced under a random interval schedule, a random interval 30-second schedule. So we always have 2 reinforcements per minute, but we're selecting short inner response times, or high rates of behavior. This is called a cumulative record. Here we're looking at cumulative responses under a multiple schedule in which we have percentile alternating with DR8 and percentile and DR8. This is what the behavior looks like for a particular animal. Under the DR8 schedule, this is a DR8 of 9-4, so 9 responses within 4 seconds. We get this nice pattern of bouts, if you will, of responding. We'll get 9 responses, criterion, 9 responses, criterion, 9 responses, criterion. These slash marks actually show criterion responses, and down here we see where the reinforcers are delivered. This really, I think, is a second order random interval of 30 seconds of a percentile. Look back under a 10, reinforcing 50% of the previous. The attitude was shorter than 50%. And this is what the percentile looks like exactly after the response. So the 10 advances for each response. It ticks for each criterion responses. Exactly half of the responses are criterion responses. And we get reinforcers under a random interval schedule. So we're comparing these 2 procedures. And one of the things that needs to be explained, and I'll address the explanation a little bit later in the talk, is a phenomenon behavior that looks like this. So this shows the acquisition, group averages across rats exposed to no mercury. This is chronic adult onset exposure that we know produces motor deficits. No mercury, quite a low dose of mercury and a moderate dose of mercury. And we see that overall response rates are higher in the control animals than they are in the exposed animals. But the question is, what's the behavioral mechanism? Why is this happening? And we're hoping that we can identify this. And we're hoping that we can do this by feeding the inner response times that arise from these cumulative records into a machine that spits out parameters that describe the survival plot of inner response times. So this shows a log survival plot. It comes straight from Rick Schall's work with that breaks responding into the behavioral stream into bouts of IRT. So to walk you through this one, this event record shows a bout of responding. Each vertical line represents a lever press, separated by a period between bouts, a bout of responding, a period between bouts. The inner response times within this bout form the declining pattern of IRTs that are very short. And you can fit this particular function with an exponential curve. And this describes IRTs in this particular case that are less than, I think, about a half a second. A parameter from this function, 3.5, describes the within bout response rate, which in this case is 3.5 responses per second. It's about high rate responding. The interbout intervals form this leg of this plot, which we normally do a log coordinate, but I'm going to do it here. And the interbout interval, and the parameter that describes this is 0.11, which means that we get, about 0.11 bouts per second. one value reaching 9 seconds per second. The y-intercept here shows is related to the Bout length. I'll be happy to walk through y, but in the interest of time, you may have to take my word for it. In this particular case, 1 over it works out about 9 responses per Bout. I think it's quite long here. Oh well, sorry about that. So the idea here is that we partition the response stream into Bouts, into parameters that describe within Bout responding, Bout initiation rates, and Bout length. And Michelle has shown fairly nicely that the Bout initiation rate seems to be very sensitive to motivation or incentive measures. This is appealing. It suggests that schedule type variables that affect response rate will affect the rate within a Bout. This too was appealing, but we were also thinking that motor deficits might also affect the within Bout response rate. And motor deficits, well, actually, in retrospect, we now know that motor deficits can affect all of these. I'll talk about that in a moment. But this can tell us some of the mechanisms, I think, by which motor deficits can exert their influence. So, this work was conducted with VI, very similar to random interval schedules. It turns out it seems to depend on that. And so as we brought this into the laboratory, we wanted to get a feel for it. I think it's important in applying a model to tinker with it for a while. Does it do what you think you're doing? Does it work in your particular application? And our particular application is actually quite different from this because we were looking at percentile schedules and high rate responding and it's very different from that. So one of the things that we did was we varied the proportion of inner response times that feed into this selection criteria. And so if we require that the current inner response time be shorter than half of the previous, in this case 10, than half the previous 10, then we get an average within Bout response rate of slightly greater than 2. And as we go up to requiring that it be shorter than 90% of them, then we increase our within Bout response rate. That's nice and makes sense and exactly what we expected. The important thing, though, too, is that this was independent. So it had no impact whatsoever on the initiation rate, which seems to be sensitive to other things. I believe seems to be sensitive to motivation variables. Bout length was sensitive also to this, but only at the 90% point. We compared, you did most of this, percentile reinforcement of short IRTs with random interval schedules, and with the eye looking at a number of different variables. Let's see, that's not the one I wanted to show. That's nothing wrong with it. I want to combine two here. So we looked at overall response rates and overall reinforcement rates. With a percentile schedule, we were able to increase the overall rate of responding. These distributions represent a range of overall response rates, including some sessions that occurred early in training. We wanted to get a full distribution, so we included a broad range of sessions. The RI schedule maintained overall rates of about 25-30 responses per minute in this particular arrangement. However, the overall reinforcement rate was not changed. So the RI schedule, the random interval schedule, did exactly what a random interval schedule is supposed to do. Our within Bout response rates were also affected with our percentile schedule, which we reinforce short IRTs. We get within Bout response rates in the order of 3 responses per second. Again, these data come from a range of conditions, including early in acquisition. And our RI schedule maintains slower within Bout response rates. Bout initiation rates were relatively unaffected, although there's some variability, especially early in training. Okay. Our Bout parameters are correlated with response rate. We wanted to demonstrate this, so we did it this way. So these show scatter plots. Each color represents a different graph, and we're looking at the correlation between within Bout response rate ranging up to about 6 responses per second and overall rate of reinforcement. That correlation is about 0.6, taking over all of these subjects. And a number of different phases of the experiment. The bout length is also correlated, so we get bout lengths that go up to as long as 60 or 70 in some cases, and that's correlated with overall rate of reinforcement. The bout initiation rate is correlated with overall rate of reinforcement. Overall rate of responding. But fortunately, the overall rate of reinforcement and the overall rate of responding did not show much of a correlation, so we were able to target this high rate behavior while holding our reinforcement rate fairly constant. That is at all positive because in some sections, of course, there are very low response rates. Bout parameters are weakly correlated with each other. So these show the correlation that I just told you about, 26.459. But within bout response rate and bout initiation rate, within bout response rate and bout length, and all of these different bout parameters are fairly weakly correlated with each other. It disturbed me a little bit that they were correlated at all, but we can live with this. So they are related to overall rate of reinforcement, which is good. They are not related to each other, but suggest that they give us some independent measures, some independent descriptors of behavior. And so the next question is, can we actually predict or account for or have the overall response rate with these parameters? And in fact, we can. So if we look, so what we're doing here is looking at a range of actual response rates going from very low up to fairly high, and we're predicting it with this equation. These are normalized coefficients, so we can see the relative importance of these different bout parameters in predicting overall rate of reinforcement. And we can account for about 90% of the variability across a number of animals by looking at bout length, log-scale bout length, initiation rate within bout response rate, and a little bit of contribution from those reinforcement costs. Okay, taking it to the streets. So now, can we perturb behavior in a way that, can we perturb behavior with a challenge, in this particular case a drug challenge, and can we account or describe these perturbations of behavior with this approach? Pentobarbital is a wonderful drug for studying this type of behavior. Dating back to Peter Dewey's mid to late 50s, pentobarbital has been shown to elevate, at moderate doses, the rate of responding that is maintained by ratio of type schedules of reinforcement, or elevate high rate behavior. So we came in with our percentile and our GRA schedules. We administered 50 doses of pentobarbital. This is a complicated graph. I want you to just pay attention to the dietary sufficient condition in the animals that are not exposed to methylmercury. You see filled circles in the bottom. You're welcome to look at the other graphs if you like. It may just confuse you, but we do have mercury interactions that I really talk about. How we did. So we get a nice elevation in the overall rate of responding. We're looking at responding at a percentage of control with pentobarbital, so that at about 5.6 to 10 milligrams per kilogram, we get 50 to 75% increase in overall response rate equal to mean. And then as we push the dose, we basically wipe out responding. Well, pentobarbital has this effect on high rate behavior, but it also, it's like alcohol. It swamps the cerebellum, it produces motor deficits. It makes it difficult to respond. So can we distinguish the things that contribute to the high rate, to the rate increases, from those things that contribute to the rate decreases? So here we've looked at, again, just pay attention to the filled circles in the bottom graph. So here we're looking at bout initiation rates, and within bout response rates, and we're looking at dose effect curves, pentobarbital on this phenomenon, on these endpoints. And we get rate increases in the bout initiation rate up until we completely eliminate behavior. It looks like a 2.5-fold increase in the rate in which bouts are initiated. Within bout response rates respond differently. It's monotonic, not bitonic. No, not bitonic either. But we begin to see the motor deficits that occur, and that seem to be independent and separable from the rate increases that we see with pentobarbital. So we can perturb this behavior, and this model, this nice descriptor of behavior helps us understand what these perturbations mean. We are also able to detect effects of methylmercury, and I won't have much time to go into this, but this is one of those things that's making me go back and re-look at what these Bout parameters may be telling us. So here we look at methylmercury. We know that I've already shown you that methylmercury exposure reduces overall responding on these high rate procedures. We're looking here after 200 days of age, up to about 100 days of exposure, chronic exposure. We're looking at the Bout initiation rate. Let's see, that's the wrong slide. I'm sorry, within Bout response rate, and the Bout initiation rate. Within Bout response rate correlates about 0.51 with total response rate, initiation rate about 0.63, so we're capturing something with overall response rate. With chronic exposure to methylmercury, we see a pretty substantial decline in overall response rates as a response per minute. And then at this high dose, where we begin to really see some pretty significant motor deficits, we also see some deficits in Bout initiation rates. This puzzled me a little bit, but then it occurred to me. Bout initiation rate not only is a motivational, properly motivational to some extent, and that makes it sort of like a pre-ratio pause, which also is driven in part by the upcoming ratio and has some motivational components to it as well. I think what this is telling us is that there's motor deficits going on, to be sure. I won't talk about nutrition interactions. They are interesting, but a topic for another day. So we'll just look at the deprived diet here. But I think that this figure is also telling us that with an impaired animal, undertaking a bout of high rate responding is difficult. It's like a high ratio. And so overall changes in response rate are driven not just by motor characteristics, but also by how difficult the procedure is. We found a problem with fitting these curves, especially with the DRH schedule. I won't have a chance. I didn't think I was going to have time to talk about it, but I'll skip over that. In summary, it's time to start thinking about translating our results into other domains. Good models reduce behavior to fundamental elements, and this should allow us to forge links to studies that are conducted at different neurobiological levels. It should allow us to talk sensibly to other people about effects that perturbations in neuroanatomy, perturbations in neurochemistry, perturbations in genetic pathways or in biochemical pathways have at the level of behavior. Among the criteria for extension in thinking about translating a model or extending it to another arena, take special care, look very closely at the application and related to the validity, related to what the model is telling you. Be sure, check for the presence of interpretable, understandable parameters that make close contact with behavior, most close contact with independent variables that you're looking at. And if necessary, and I think it is, as we start entering the world of behavior genetics and high throughput issues, start thinking about issues of scalability. There are a lot of people in this room who like the sorts of programs and scale up in the ways that we do, that I do, with great facility. And I think we need to bring that skill, make that skill more broadly available. Thank you for your attention. I'll be happy to take any questions if there are time.